facing or touching the afferent arteriole, the cells over there become columnar, they become thick, and they become dark, they look dark, and that is called macula densa. Macula is spot, densa, dense, a dense spot. Then of course between these cells are small mesangial cells here as well, or the interstitial cells. We are less concerned about those mesangial cells, we are more concerned about this structure. The same structure, if I draw here, it will be macula densa cells here and the pericytes here. Afferent arteriole, pericytes, and then the distal convoluted tubule, and the cells of the distal convoluted tubule. The only difference is here we are talking about the cells of the distal convoluted tubule wall. Here we are talking about the cells which are surrounding the arterioles. Very important concept. I'm not wasting time here. This is such an important concept that you would see so many questions and you would see so many practical applications of this, that if you do not know this, you would be at a loss. So please do not forget this, write it down somewhere. These pericytes are also called granular cells. These are also called JG cells. These are also called modified smooth muscle cells. These are all the four things the same thing. The pericyte or modified smooth muscle cell or the granular cell or the JG cells are the same thing. Cool? These are the cells outside the arteriole. Then macula densa cells are the columnar cells present in the distal convoluted tubule. Now here let's talk about the function. That is how we'll connect it back to the osmolarity and the blood pressure. So I had said to hold that thought, so time has come to go back to it. What is in the distal convoluted tubule? Again, unfair thing from my side, I have not taught the nephron yet. So I'm not expecting you to actually know what is in there, but I'm asking you this question. Inside the nephron, so inside this nephron, what is happening is blood is coming into the glomerulus. From here some filtration would occur and nephron would have urine in it. That is the waste products which we want to throw out. That is the function of the kidney. So nephron is the one which is actually performing the function. We bring the blood in here, we squeeze the blood into these nephronal structures. Nephron's function is then to process that blood, form the urine out of it and throw the urine out. So I can tell you that when the fluids and substances pass through the distal convoluted tubule, they are almost like urine. Not fully urine, but they're almost like urine. So may I say that the substances which are passing from here, I wish I had a yellow marker, the substances which are passing through here are urine. Similarly, whatever is present here is urine. On the other hand, what is present in the afferent arteriole? Blood, right? What is going into the afferent arteriole? Blood. What is coming into the afferent arteriole? Blood. So on one end we got the blood which is passing through the arteriole and the pericytes are present around the arteriole. On the other end we've got an urine present in the distal convoluted tubule. Cool? Next concept. Attached to this concept, pericytes have stretch receptors. They are blood pressure apparatus. Do you know that we have 1 million nephrons per kidney? And every nephron and the glomerulus in it has one blood pressure apparatus. So blood pressure, what is in the blood vessel? Blood. And what is around the blood vessel? It is a cuff of the cells. Just like you as a doctor or a medical student or a nursing student, you put on the cuff around the patient's arm and you take their blood pressure, right? This is the same cuff over here. It is a biological blood pressure apparatus. How many apparatuses do you have? You have 2 million blood pressure apparatuses in the kidneys. Very interesting phenomena. You have 2 million of those. So one of them is here, what you're seeing. So these pericytes are monitoring the blood pressure inside the arteriole. So when the blood pressure is more, the arteriole stretches out, the pericytes would stretch out, and they'll say, oh my god, the blood pressure is more. On the other hand, when the blood pressure is less, these would collapse and the pericytes would say, oh, the blood pressure is less. On the other hand, the distal convoluted tubule, does it has to do anything with the blood? No. It's a tubule which has urine in it. Can it measure blood pressure? No. What does it measure? It would measure whatever is present in it. What is present in it? Urine. So what this thing measures is the concentration of sodium here. So can I say now, this is a chemical sensor, the macula densa, and the pericytes are baro sensors or receptors, that is pressure receptors. So not only you gotten 2 million blood pressure apparatuses, you also got 2 million chemical apparatuses which would be looking at the chemical composition or osmolarity of the substances passing in the urine. Good. So this is done as well. What are the cells here? JG cells and macula densa cells. Now let's see how do they function. How do they help maintain the osmolarity? So this is what happens. When the blood pressure is more, pericytes get stretched, and when pericytes get stretched, these will release renin. These would release renin into this area. Renin is released by these cells, and I hope you understand what happens is liver makes angiotensinogen, which is a precursor for angiotensin. Renin is the enzyme which would come in and convert angiotensinogen to angiotensin 1. Angiotensin 1 then circulates in the body, and in the past we used to think that only the lungs and the endothelium of the lungs, or some cells in the lungs, were responsible to convert angiotensin 1 to angiotensin 2. Now we know that all of the endothelial cells in the body are responsible. They have ACE or angiotensin converting enzyme. That enzyme acts on angiotensin 1 and converts the angiotensin 1 to angiotensin 2. Cool? So now we got in renin. Renin is going to work on tensinogen, which is coming from liver. Liver makes that. Liver makes most of the proteins, so there is no mystery here. Renin is going to work on the angiotensinogen, which would make angiotensin 1. Angiotensin 1 will be acted on by ACE, angiotensin converting enzyme, which will convert it into angiotensin 2. This angiotensin 2 is responsible for many things in the body, but one important thing which it does is it converts. One important thing which it does is that it causes vasoconstriction. In this particular case, it causes the vasoconstriction of efferent arterioles. And we'll talk about it, that when the efferent arteriole is constricted, there will be more glomerular filtration. That filtration would then cause more of the GFR and those things. Plus, because angiotensin 2 is a vasoconstrictor, it would increase the blood pressure. It also goes and acts on the pituitary, releases the adrenocorticotropic hormones, which goes to the adrenal glands, and release aldosterone. Aldosterone in turn cause the reabsorption of sodium, which would cause reabsorption of water as well, and that would increase the osmolarity plus the blood volume. Again, the point important here is to note that these JG cells or granular cells or pericytes or the smooth muscle cells, modified smooth muscle cells around the arteriole, efferent arteriole, their function is to look at the blood pressure, and if they feel that the blood pressure is going down, then they would release renin, and renin's job is to bring together angiotensin 2, whose job is to increase the blood pressure. We'll talk more about it. So it's not finished yet. This is just a part of the concept. The second thing now, so that is the blood pressure part, sensor part, and the function. Now look at the macula densa. Macula densa macula densa receives the sodium, and that sodium